You will remember again, even if you have Alzheimer's. In due time, this could be a reality with results from Cognition Therapeutics' Shimmer and Shine studies showing promise, suggesting that zervamecine may be an effective treatment for patients with DLB and Alzheimer's disease. Cognition Therapeutics recently presented results from 2 phase 2 clinical trials of zervamecine, also known as CT1812, at the Alzheimer's Association International Conference, AIC 2025. The first trial, called Shimmer, focused on patients with dementia with Lewy bodies, DLB, a neurodegenerative disorder that affects cognitive function, motor skills, and behavior. In this study, zervamecine demonstrated a positive impact on various aspects of DLB symptoms. Patients treated with zervamecine for 6 months showed significant improvement in neuropsychiatric symptoms, such as hallucinations, delusions, and anxiety, compared to those receiving a placebo. According to Dr. James E. Galvin, the director of the Comprehensive Center for Brain Health at the University of Miami Miller School of Medicine and the Shimmer study director, these symptoms are often the most troubling for patients and their caregivers and are challenging to treat due to severe reactions to commonly used neuropsychiatric medications. Notably, patients treated with zervamecine scored an average of 86% better than placebo-treated patients on the Neuropsychiatric Inventory, NPI-12, a tool that measures 12 separate behavioral symptoms. The second trial, called Shine, investigated the effects of zervamecine in patients with Alzheimer's disease. This study met its primary endpoint of safety and tolerability, and further analysis revealed that patients with lower levels of plasma p-tau217, a protein associated with Alzheimer's disease pathology, responded more robustly to zervamecine treatment. According to Dr. Anthony Caggiano, Cognition's chief medical officer and head of R&D, this finding is significant because it suggests that a simple blood test for p-tau217 can identify patients who are more likely to benefit from zervamecine treatment. In these patients, zervamecine was shown to arrest further cognitive deterioration by 129% in those with mild Alzheimer's disease and 91% in those with moderate Alzheimer's disease. The SHINE study also explored the impact of zervamecine on various biomarkers associated with Alzheimer's disease biology. The analysis of cerebrospinal fluid, CSF, and plasma biomarkers revealed significant reductions in plasma glial fibrillary acidic protein, GFAP, a protein associated with neuroinflammation. Additionally, there were trends towards normalization of neurofilament light, NFL, a protein associated with neurodegeneration, and amyloid beta species, A-beta 40 and 42. According to Dr. Mary Hamby, VP of Research, these findings suggest that zervamecine treatment affects proteins involved in neuroinflammation, synapse health, and neurodegeneration, which is consistent with previously reported results. This builds on earlier progress from results published about proteomics analysis from the Phase 2 sequel study of zervamecine in adults with mild to moderate Alzheimer's disease in the peer-reviewed journal Alzheimer's and Dementia, Translational Research and Clinical Interventions. The sequel study investigated the effect of zervamecine on synaptic function using quantitative electroencephalography, QEEG, a non-invasive technique to measure electrical activity of the brain. The study found that zervamecine treatment resulted in consistent trends of improvement across all pre-specified EEG parameters, including the prominence of certain EEG frequencies associated with Alzheimer's disease. Specifically, zervamecine treatment reduced slower frequencies termed theta waves after 29 days of treatment compared to placebo, suggesting that zervamecine may normalize the function of neurons and synapses. Additionally, zervamecine treatment was associated with an improvement in global alpha AECC, a measure of functional connectivity, suggesting that zervamecine may be facilitating communication between brain regions. Analysis of proteins associated with zervamecine's normalizing effect on AECC revealed that proteins significantly altered with zervamecine treatment compared to placebo were associated with vesicle formation, exocytosis, and endosomal trafficking. These key cellular functions allow neurons to recycle and eject cellular debris and transport cargo needed for cellular function. According to Mary Hamby, Ph.D., Cognition Therapeutics VP of Research, these findings suggest that zervamecine has the ability to preserve the health and function of neurons that are under stress in Alzheimer's disease. To further investigate the neuroprotective function of zervamecine, researchers conducted a series of in vitro experiments using neurons derived from a human cell line. These neurons were exposed to 4-hydroxynonenal, 4-HNE, a naturally occurring molecule known to induce oxidative stress, which is found in high levels in the brains of people with neurodegenerative diseases. When added to a culture of neurons, 4-HNE caused cell death, evident by the visual appearance of the culture and an increase in levels of neurofilament light, NFL, released from dying neurons into the culture medium. However, when zervamecine was added with 4-HNE to the cultures, cell viability was preserved and cellular levels of NFL were maintained, attesting to the structural integrity of the neurons. Further studies showed that zervamecine was able to preserve cell integrity and function by interrupting 4-HNE's ability to induce cell death. Dr. Hamby noted that additional research will be needed to determine the precise point at which zervamecine prevents cell death under oxidative stress conditions. This neuroprotective mechanism may underlie the decrease in NFL observed in Alzheimer's patients treated with zervamecine in the 6-month SHINE study of mild to moderate Alzheimer's disease. To sum it all up, the take-home message is that these findings suggest that zervamecine may have a neuroprotective effect, preserving the health and function of neurons under stress in Alzheimer's disease, and support further investigation into its potential as a treatment for neurodegenerative disorders.